Okay, thank you for the introduction. Um, Biological systems are intricately spatially organized. Uh, right now, we have tools such as bulk and single cell sequencing. These can show us parts of a biological system, but they can't show us the entire picture. They can give us information on a tissue level as to what is going on at the tissue level, but they can't show us how cells are spatially organized. And so we here at Vizgen, we want to move towards providing spatial genomics with true single cell resolution. We feel that this offers a higher level of insight in order to look at very tough, complex biological systems. And so in order for us to do that here at Vizgen, we have developed the MERSCOPE platform. This is a comprehensive platform that allows users to run hundreds to thousands of genes in a single run. We can detect transcripts from all of those genes. It's highly sensitive and accurate. We boast a large imaging area of 1 square centimeter and a resolution of 100 nanometers. And so what this gives you the ability to do is look at an entire tissue section, look at different regions, and then zoom into specific areas and look at how cells are interacting, what those cells are doing, and how they're functioning. There's no need for sequencing with our system as it is based on our proprietary MERFISH technology, which I'll get into in a second. And in addition to being a comprehensive system, we've also built in a user-friendly aspect. So our system comes with control software and visualization software that allows users to look at their data and visually identify different items within that data, different data sets, all of this with an intuitive system. In addition to that, we offer a web-based gene panel application. This allows users to build gene panels for their specific research, build those custom panels within their application areas. And we see a wide range of application areas being used or being studied with our system. And so we're very excited to offer these features. And so I mentioned our system is based on MERFISH. That stands for multiplexed error robust fluorescent in situ hybridization. This is a proprietary technology. It is unique to Vizgen. MERFISH is built on SMFISH. SMFISH allows direct quantification of RNA targets within tissue. We combine that with our proprietary barcoding system that allows us to build these gene panels to target more than 500 gene targets and detect all of those transcripts within a single experiment. We combine SMFISH with our proprietary barcoding system. What this gets us is a highly multiplexed RNA detection system called MERFISH that enables single cell analysis. The error robustness of MERFISH allows for that high accuracy and consistency we need for investigating spatial genomics data. We combine all of that in an easy to use, fully automated platform. And this platform allows researchers to gain new insights into complex biological systems. Again, MERSCOPE is the only proprietary platform for MERFISH technology. It includes not just the instrument itself, but also reagents, control software, and analysis software to provide an end-to-end solution with an exceptional range of application, interests, and industries. All of these tools come together to offer a end-to-end system that makes studying spatial biology easy for users. And so if we take a look at what that data starts to look like, this is an example data set of a mouse brain. Here we're showing 8 RNA subsets. So we've highlighted 8 genes and looked at the transcripts from all 8 of these genes. And what you can start to see across the tissue section is different regions of the tissue lighting up with different RNA transcripts. And so what this allows you to do is look at these areas of the tissue, look at how they're interacting, what cells potentially are in those areas. And then in addition to this, we can zoom in. And in this bubble, we show a 200 by 200 micron zoom in, and you can see that individual cells here are lit up with different genes, and you can start to see how they interact, which genes look different across different cells, and really get insight, valuable insight into what is going on not just a tissue level, but on a cellular level and potentially subcellular level as well. What a big focus for us here at Vizgen is to continue to expand MERSCOPE applications. We know that the MERSCOPE platform offers the ability to detect transcripts from 500 genes. It offers that subcellular resolution, but we want to continue to push forward in developing product development and enhancements for the MERSCOPE device. The first one, major focus area for us, was in expanding the analytes detected. We want to move towards a multi-omic measurement that includes different types of analytes, as we know that transcripts alone don't tell you all of the information on how a cell is behaving. And so that moved us towards developing the MERSCOPE protein co-detection solution. The second major area we wanted to move into was enabling a diverse range of samples and unlocking those archival samples, especially FFPE tissue samples. We know that FFPE is a very common tissue preservation technique, and so that moved us towards developing the MERSCOPE FFPE solution. So now in these next few slides, I'll go through the MERSCOPE protein co-detection solution, followed by applications, and then jump into the MERSCOPE FFPE solution. Okay, so if we take a look at where we were with the MERSCOPE device about a month ago, we had the ability to look at just transcripts alone, right? And unfortunately, we know that cellular transcriptomes provide an impartial picture. They don't capture what's going on a protein level. And as a result, the cellular state and function cannot be fully captured by a transcription profile alone. Additionally, we know that post-translational modification downstream of transcription of certain genes is important and has been implicated in certain disease states. On top of that, we know that protein-based cell labeling, it's common within certain fields, especially oncology. And here at Vizgen, what we want to do is enable researchers to build upon existing information, build upon existing techniques. And so at that time, we only had the ability to detect RNA alone. So through product development, we wanted to get to a point where we could provide researchers the ability to generate multi-omic data, detect both RNA and protein at a subcellular level. Through our product development, we accomplished this through oligo-conjugated secondary anti-species antibodies. How this works on a technical level, we have protein target outlined in gray here, and this can be a target that a researcher is interested in, and they have a primary antibody. Most researchers will have a primary antibody they've been using, and the beauty of this system is that it employs that primary antibody provided by a researcher. So you don't need to go back to square one and revalidate binding of a new antibody. And so in this example, we have a protein target that is being targeted by a primary antibody from a host species. For instance, we'll say a mouse. Here we would use our anti-mouse oligo-conjugated secondary antibody. This antibody will bind to the primary antibody, and what it has is an oligo conjugated to the back end of it. This oligo contains an encoding probe with a detection probe binding site. When we get to the round of detection, the complementary sequence on that detection probe will bind to that detection probe binding site on the back end of the oligo, and the fluorophore, which is attached to the detection probe, will then show signal that is proportional to the presence of the protein. How we've package this is we offer protein stain kits. So those antibodies are contained within these protein stain kits. We offer 6 different types, so an anti-mouse, an anti-rabbit, anti-goat, rat, human, and chicken. Each one of these protein stain kits is good for 20 samples, 20 MERSCOPE samples, or verification samples, which I'll get into in a second. It also contains the blocking buffer that is required in order to move through the workflow here. Now, important for us here at Vizgen is we want to offer researchers the ability to not just do the initial experiment, but do a verification so that they can verify and optimize their sample prep process before actually going into a full protein co-detection experiment. We have a similar setup with our sample verification kits that allow researchers to do an initial verification. And so here, what we also developed in addition to the protein stain kits is protein stain verification kits. These kits contain the reagents required to run through the protein stain verification, which is a shorter overall protocol that allows users to first test that the primary antibody they're going to be using can be used in conjunction with our protein stain kits. These protein stain verification kits come in pack sizes of 5. We have 2 of these. So we have a mouse, rabbit, and goat protein stain verification kit and a rat, human, and chicken verification kit. You can do you can do verification of 1 antibody or 2 or 3. They just have to be within this grouping. So in a similar sense here, we're giving researchers a way to first verify before moving into a full experiment, which is important. Additionally, as part of this solution, we also introduced a couple workflows. So we have a protein stain verification workflow. Again, this is where we're going to verify that the antibodies we're going to be used are going to work. And then we have a workflow for the full MERFISH protein co-detection. Now, what I have on the screen here is what materials are required in order to move through those protocols. And so in the blue table, we have the materials that are required for the protein verification workflow. So here the researcher would be responsible for providing the primary antibody or antibodies. You would need your MERSCOPE slide box, which contains those slides where you can mount the tissue that you're going to do the testing on. The MERSCOPE sample prep kit, which contains all of the buffers, reagents needed to process that sample to open up the analytes for detection, along with the the protein stain kits. Again, these contain the secondary oligo-conjugated antibodies we would use to detect those primary antibodies. Additionally here, since we're not doing a full MERFISH run, you would need the protein stain verification kit. So we have 2 variations of this protocol depending on the antibodies you're going to be running. If we look at the pink table, we can see the required materials for protein staining within MERFISH. So this is true protein co-detection with MERFISH detection as well. So we're going to detect transcripts and proteins together. Here you would need your primary antibodies, your MERSCOPE slide box again, your prep kit. You can plex up to 6 antibodies per experiment. You just need to have different host species primary antibodies. This is where you would use your gene panel, because now we are running full MERFISH, your gene imaging kit that matches that panel size, and your cell boundary staining kit. This is an optional item, of course. The only caveat here is if you use a cell boundary staining kit, you cannot use it with the anti-rabbit protein stain kit. Future development on this product will hopefully eliminate this compatibility issue. The other great thing about this product is that it is a very simple workflow. And so if we look first at our protein stain verification protocol, we're going to verify that the protein stain is working properly. Those of you familiar with our standard sample prep protocol, this will look very familiar. So we have a sectioning, fixing, and perming that's getting the sample ready for detection of the analytes. In this case, it's protein staining. For the actual protein staining itself, This is a high-level overview of it. Very simple. It consists of multiple washes with a blocking and incubation step, a primary antibody stain and incubation step, and a secondary antibody stain and incubation step there. Overall, very simple protocol. You do those incubations, you do the washes, and then following this, you move back into our standard sample prep protocol of gel embedding and tissue clearing, which are important for us and something that we feel very confident that provides a way to clear the tissue and get the most accurate data possible. We would then go into a readout probe hybridization, pre-imaging staining, and imaging from here. Following this, we would look at the images generated within our MERSCOPE Vizualizer system. From here, if the sample is oversaturated, we would potentially lower the power settings within the control software. These are power settings that are easy to access while you set up your experiment. Or you could also adjust the primary stain concentration. Right? So in this way, this protein stain verification protocol allows us to optimize things before we move into full protein co-detection. Once we've done that, we move into the protein co-detection protocol. Here, very similar again. We have sectioning, fixing, perming, and then we go into cell boundary staining and protein staining. It was important here that we provide the ability to do one or the other or both, right? The beauty of the system is that if you are going to do both, you do not have to do double the time. What you're going to do is actually make a cocktail of reagents. And again, both of these, this entire step here is completely optional. So if you're going to do cell boundary staining, you're going to follow the same blocking, primary antibody stain, secondary antibody stain. Similar if you're doing only protein staining, right? Different blocking buffer that comes with the protein stain, that would be the difference here. Then go through your primary antibody stain and secondary antibody stain. It's similar setup to our protein verification kit. If you want to do both, you're just going to combine both reagents into a single cocktail that is then going to be incubated on the sample. And so again, very simple protocol. It provides flexibility and allows you to generate that multi-omic data. From this step, you move through our standard MERFISH probe hybridization step. That's where you would incubate with your gene panel, gel embedding and tissue clearing, again, in order to clear the tissue and generate that high accurate data detection of those fluors, and then a pretreatment with DAPI and poly-T and imaging. So now that I've kind of gone over how the product works and the general concept behind it, I did want to show some data and some applications on how you could leverage some of this protein co-detection data. And so the first example here, we have U2OS cell culture samples. And here we wanted to show how you can use and leverage the protein co-detection to look at interrogate subcellular functions. Right? So here we've run a gene panel of 130 genes. We detect transcripts from those 130 genes together with a 5-plex protein stain. And if we start to look at that high magnification in the top right image, you can see how we can use our visualizer system to look at the protein staining together with the transcript staining. Taking those high magnification images and looking at within those cells, we can start to understand how transcripts are interacting and where they're potentially spatially organized here. For instance, fibrillin-2, we look at the transcripts, which are shown in white. We look at LAMP1, GRASP2, actin, and HSP60. We can see that it is co-localized with all of those protein markers. It is not co-localized with SOX9. What this pushes us to infer from this is that this fibrillin-2 transcript is co-localized with cytoplasmic protein markers and implicated potentially in cytoplasmic regulation. MALAT1, on the other hand, if we look at this, is very different than fibrillin 2. MALAT1 does not co-localize with those cytoplasmic markers, but does co-localize with SOX9. SOX9 is a nuclear marker, and so we can infer from this that MALAT1 is potentially regulating nuclear signal. In addition to U2OS, we wanted to look at a potentially clinical sample, so fresh frozen human colon cancer. And here what we want to demonstrate is the power of the protein co-detection to further antitype cell types. With this panel, this panel of 244 genes, we detected over 35,000 total transcripts. Additionally, we looked at 5 protein stains. These protein stains were mostly geared towards proliferation markers and cancer markers. And so when we look at that, we see pan-CK, HER2, Ki67, and VIM are all those cancer marker and proliferation markers. We see our standard expected spatial patterning here. What we can further do from here is take a look at, use these protein markers to further annotype these cell types. And so we can use transcript data. From that, we can do cell clustering analysis and generate these cell clusters based on which genes cells are expressing. We then can take those cells and annotype them based on their spatial clustering, right, where they're spatially located. From there, if we pull in the protein staining, we can then say these specific cell types are located within this area, and using the protein markers for those specific cell types, further subtype some of these cells. So look at where fibroblasts are, and are there a population of fibroblasts that are potentially located in a different area and expressing a different subset of genes, right? And so we can do this not just for the immune cells, but for the cancer cells as well. And what this gives us is the ability to further annotype these cells using the protein staining. And so the next major product I wanted to focus on is our MERSCOPE formalin-fixed paraffin-embedded solution. So if we look at tissue preservation today, there's a couple different major types of preserving tissue. For DNA and RNA and sequencing analysis, fresh or fixed frozen sample preservation is preferred. These samples are usually stored at -80. They're often not available for clinical practices, but they do present compatibility with single cell sequencing analysis. However, we know that FFPE samples, formalin-fixed paraffin-embedded samples, are the most widely used clinical sample type, right? Especially within oncology. This is a very common sample preservation system. As these samples can be stored at room temperature, they're also very good at preserving tissue morphology. The problem here is that it cross-links the tissue, it degrades it, and can oftentimes fragment RNA transcripts. So even though we have a great deal of archival samples stored as this, it presents a problem in trying to analyze this and get at that RNA within the tissue. So in order for us to open this tissue up for spatial analysis, we developed the MERSCOPE FFPE product solution. And this solution is composed of 3 major pieces and components. The first is reagents. So we have new kits that provide reagents necessary to enable our new FFPE sample prep protocol. So this includes our sample prep kit, our FFPE sample prep kit, and our FFPE slide box. The second major piece of this is our new protocol. And this is going to allow us to unlock those FFPE samples, open them up, expose those analytes, not just transcripts, but proteins as well, and analysis, and open those samples up for downstream MERFISH analysis. The final major component is software. And so with this product release, we will be offering a new cell segmentation offering, which is based on CellPose, which provides an improved cell segmentation analysis for tissue samples. Now I'll kind of go through each one of these. So the first major component, as I mentioned, was our FFPE specific MERSCOPE reagents. So here we are releasing an FFPE sample prep kit. This is going to contain those new reagents needed to unlock those FFPE tissue samples. Together with that new sample prep kit, we are also going to be offering a new FFPE slide box. This is going to contain new slides, new FFPE specific MERSCOPE slides that are going to be compatible with our FFPE sample prep kit. If we start to look at how the portfolio looks on the side here within this table, we now have different reagents for different sample types. Right, so for your fresh frozen and fixed frozen and cell culture samples, you will use our standard sample prep kit. There are two flavors of our slide boxes for these. Our cell culture system requires non-beaded slides in order for researchers to grow the cell culture on the actual slide itself. From here, you use our standard cell boundary staining kits, our protein stain kits, our gene panel, and our gene imaging kit. Now with FFPE, we've offered another route for these samples. You will use our FFPE slide box, our FFPE sample prep kit, along with those common reagents. Again, I want to stress that our new protein stain kits, which I just went over, they are compatible with our FFPE sample prep processing. So very important, very powerful. But now we have offered all of these different reagents that researchers can use based on the sample they want to investigate. So now the second major component of our FFPE sample prep solution is our FFPE protocol. So here we had to kind of change things up because we know FFPE samples, they have that wax, they have that cross-link, they cross-link the tissue. So we needed to develop a new protocol in order to open the tissue up, expose those analytes for downstream MERFISH imaging. So moving through this protocol, the first piece is sectioning. So we're going to section a slice off of an FFPE block. We would then mount that on our MERSCOPE slide. From here, we would move into deparaffinization. It's a chemical and temperature process to remove the wax from the tissue. We then do a decross-linking procedure to remove those formalin cross-links and further improve access to the analytes. We follow this with an anchoring pretreatment, which improves the RNA binding to the gel. In the next few steps here, we'll have gel embedding, but this is an initial treatment to improve that binding. Here, again, I want to stress that we have secondary staining. This is where we can use that protein staining within the FFPE workflow to get and generate that multi-omic data. So here, this is completely optional. It includes the cell boundary staining and the protein staining to mark those secondary analytes. Following this step, we move into RNA anchoring. This is a final treatment to prep the RNA for anchoring to the gel. We then move into gel embedding and clearing. Gel embedding and clearing are critical components of our protocols. We think that they help in our detection, our sensitivity, and our specificity. Right, so first we embed the gel, we anchor those RNA transcripts to the actual gel itself, and then we clear and digest all of the protein from the sample. And that lowers the background and gets us that really high sensitive signals, or highly sensitive signals. From here, we do a hybridization where we use our gene panels to hybridize our encoding probes to those RNA targets. We do a post-hybridization wash to remove the unbound encoding probes, and then we go through MERFISH imaging, which is going to flow in those detection probes that will bind to the encoding probes that are present. An optional step here is our auxiliary imaging. Here we can do further detection of, say, our cell boundary stain or our protein staining. This happens outside of MERFISH imaging, but still provides flexibility to potentially detect those proteins or detect highly abundant genes. The final piece of our FFPE sample prep solution is our cell segmentation algorithm. And so this is a new machine learning driven cell pose cell identification. It provides unrivaled processing of spatial genomics data across tissue types. We're extremely excited about this. It certainly improves segmentation within FFPE samples, where you can have these smaller cell types. It more accurately captures the cell shape in these dense tissues. It is completely integrated into the MERSCOPE user interface. So users that currently have a MERSCOPE right now, they will receive a software update that will introduce this new cell segmentation algorithm as an option, right? So we won't replace watershed segmentation that's currently available. This will be an additional option, provide more flexibility for researchers, not just for FFPE samples, although we think this is going to be very useful within FFPE segmentation, but they can start to use this segmentation across fresh frozen, fixed frozen samples as well. And so now just going through applications of our FFPE sample prep protocol here. One of the first major area we wanted to focus on within FFPE is in the quality of the data we're generating. Right? So here at Vizgen, we are very much focused on developing high quality spatial genomics data. If we take a look up top here, we're using our FFPE mouse brain sample. And what we've done is we've compared our MERFISH counts within that FFPE sample to bulk RNA sequencing. What we get is a highly correlated data, 0.88 R value, across the entire expression profile. So we're not having to insert a line that breaks this data apart between low expressing or medium expressing or high expressing genes. We get this correlation across the entire expression profile, which is very important because we know researchers are going to be interested in not just those high and medium abundant genes, but also low expressing genes as well. The second figure on the top here, we look at replicates between FFPE mouse brain samples, extremely high correlation, indicating that we are getting that reproducibility. The other place we really wanted to benchmark and generate a kind of a quality measure of our FFPE sample prep is benchmarking it against our fresh frozen protocol. And so you can see here that if we take a sample that is FFP and we take a fresh frozen sample and we compare MERFISH from both of those samples, we get a very high correlation of 0.95. We went further here to compare these and annotated these cell types here within FFP and fresh frozen samples, and then spatially compared those cell types within these two sample types. And so if we look, we can see all cells between the FFPE and fresh frozen sample. We then look at the spiny projection neurons, and you see a very similar spatial orientation for those cells, oligodendrocytes, and cortical excitatory neurons. So basically, we are seeing the same spatial distribution of these cell types within FFPE and within fresh frozen cells, which is very important. We didn't want to just focus within brain tissue. We wanted to push our FFPE system and apply this into cancer types and additional tissue types as well. What we see is high sensitivity and specificity across multiple FFPE samples. So you can see up here we have human ovarian cancer, lymph node, tonsil, human lung cancer, human skin, and human kidney. In addition to just showing that we can successfully demonstrate MERFISH here, we took these 6 samples, 6 additional FFPE tissue types, and look at the correlation coefficient. Again, looking at the data we've generated using MERFISH, comparing it to bulk RNA sequencing, we see very high correlation coefficients across all of these tissue types, which is incredibly important. And again, this is across the entire expression profile. And then finally, as I mentioned before, we want a key feature of both of these products is their ability to work together. So having the ability to do that protein co-detection within FFPE samples. And so we have a MERSCOPE FFPE. We've demonstrated this within human colon cancer FFPE sample. Here we did staining protein co-detection with 6 proteins. And so on the left, you can see pan-CK, SMA, E-cadherin. These are lit up within our visualizer. These are the protein targets. And then on the right, the distribution of all of our transcripts we detected. We detected over 67 million transcript counts. So incredible data. We look in, zooming into these areas, we can see where these proteins are differentiated, are expressed differently. Additionally, looking at transcripts, we can see how they are expressed differently as well. But again, the idea here is that we can successfully generate protein co-detection data within our within FFPE samples using our protocol. And so just very quickly, conclusion here, with the MERSCOPE, we see a lot of advantages, right? We want to continue to push the boundaries of spatial transcriptomics, not just with the platform itself, but with new application areas, new products that do that. So, we have that high cell throughput. From a flexibility standpoint, we've only improved this by providing a solution for FFPE samples. We've also, from a multiplexing standpoint, we have that ability to cover hundreds to thousands of genes using custom gene panels that you can develop. But now we are moving that multiplexing into multi-omics, right? It's not just transcripts anymore, it's protein data as well. We combine that with our resolution, and our sensitivity and specificity allows researchers to generate spatial genomics data and now multi-omic data across a wide range of samples and answer those tough, complex biological questions. So I'd be happy to take any questions at this point.